Good day mga ka-science! Ako si Ate Kim at nandito tayo sa University of Santo Tomas. So why exactly are we in UST? We are here in UST to discuss about the different innovations in science and technology. So if you want to know more, you better wear your lab gowns and tune into today's episode of Biology! For today's episode, we are going to tackle about the very controversial topic of genetically modifying human embryos. Just a few days ago, a scientist named Hejan Q has claimed to have genetically modified not one, but 2 embryos. These babies were born and were named to be Lulu and Nala. So, let's see what our dear Tomasians think about this topic.
Yes. No.
I agree because you can modify yung mga genes na pwedeng maging factor that runs within your family, na pwedeng maging factor ng magiging future diseases mo. And you will not have it.
No, because sobrang unethical niya because you're playing with something natural na hindi dapat tinatamper with.
No, I disagree because in genetic modification, we are dealing with human lives. So, yung mga tao, meron silang dignity and meron silang karapatan sa sarili nilang katawan. So, kung ang goal ng genetic engineering is to change or to alter the features, wouldn't it mean na hindi natin sila kayang tanggapin kung ano man yung kalabasan nila paglaki nila. So, I disagree because kung gusto rin naman na matanggal yung diseases or ma-prevent yung diseases, kung yung tao mamamatay talaga siya kahit baguhin niyo yung gene. So, I disagree.
Yes, but with very, very strict reservations.
I agree.
For me, no po.
So, I vote yes.
We seem to be getting mixed opinions on this. To help understand our topic better, let us ask our colleagues from the University of Santo Tomas. Let us now head to the SRL.
Dr. Heath says he altered embryos for 7 couples during fertility treatments. There has been 1 pregnancy so far. His team used in vitro fertilization to modify embryos and then applied CRISPR-Cas9 to those embryos, which aims to mutate the CCR5 gene from which the HIV latch onto to breach the cell. Which basically, as he said, when Lulu and Nana were just a single cell, it removed the doorway through which HIV enters to infect people. Healthy twin babies were born named Nana and Lulu.
I will explain to you how this procedure was done using CRISPR-Cas9. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. These are found in bacteria's DNA, which they use as mugshots for bad viruses. Cas9 is an enzyme that can cut apart DNA. Bacteria sense this Cas9 where they recognize these viruses from their mugshots.
This is the approach that they try to replicate. They start with a certain type of RNA, the guide RNA, which reads the DNA to find where the editing will take place. This RNA guides the Cas9 to the exact spot of the DNA where the cut should take place in. Cas9 then locks onto the double-stranded DNA and unzips it. This allows the guide RNA to pair up with the region of the DNA it has targeted. Cas9 creates a break in both strands of the DNA molecule.
The cell in response prepares to break. This attempt to fix the breakage might disable the gene.
CRISPR seems to be the answer to humanity's biggest problem. But is it really? To know more of this in different perspectives, we asked some of our colleagues in this matter.
I really think that this is the biggest breakthrough, not only in genetics but also in the medical field. Just think about it. You'll be able to alter not just the CCR5 gene but also the other genes which contribute to some diseases like cystic fibrosis and some other diseases. Dr. He believes that his work will ultimately benefit patients with rare diseases. This has been widely used already in other organisms such as plants and some animals, and it has shown promising effects. I think it's time that we use it on us, the ones who need it most. These diseases kill millions of people per year, and I don't think we should hold back in trying to perfect this technology. As Lin Jitong from Shenzhen Harmonicare Women's and Children's Hospital said, the important thing is that we are curing diseases through gene editing. It would help patients with rare diseases to eliminate fear, discrimination, and unfair treatment within society.
Okay, first we look at this from a scientific standpoint. CRISPR may have the occurrence of targets which could lead to cancer and some health problems. Even if we say that the gene is mutated, some still become functional proteins. Lastly, he has not yet published scientific papers to prove that the CCR5 was indeed crippled. This will also have a huge impact on the diversity and perpetuation of species. Also, from a moral perspective, this is a great violation against academic ethics and codes of conduct. Dr. Kiran Musunuru from the University of Pennsylvania said that they still have a lot of work to do to prove that the procedure is actually safe. He also said that the genes on the embryos were incomplete. The girls may not be totally immune to HIV. And there is also evidence that other genes were unintentionally edited, thus increasing the risk of obtaining cancer. He said that the babies should be born at this point in time following the use of gene editing. The procedure seems too early and too premature because a lot of science needs to be done. Dr. Hess said that a healthy child was born, but we don't know whether these babies are indeed healthy. We don't know if they would turn out to develop cancer. We don't even know if they turn out other mutations as well. Scientists are known to be risk takers, but when it comes to human life, it isn't worth the risk.
This huge announcement and discovery triggered many criticisms, not only from doctors and researchers but also people from at home and abroad. As Dr. He Jiankui said, the world has moved on to a stage for embryo and gene editing. There will be someone or somewhere out there who is doing the same thing. So let us end this episode by asking, is it really worth the risk? Or are we just spiraling ourselves to imminent downfall? Muli, I am Ate Kim, and this is Buhayology.